Continuation. Feedback inhibition occurs when a product of the Krebs cycle inhibits an earlier enzyme in the cycle. For example, ATP can inhibit citrate synthetase. Inhibitors of Krebs cycle. The Krebs cycle, also known as the citric acid cycle, is a series of chemical reactions that occur in the mitochondria of eukaryotic cells and in the cytoplasm of prokaryotic cells. The cycle is an important part of cellular respiration, which is the process by which cells generate energy in the form of ATP. The Krebs cycle involves the breakdown of acetyl-CoA molecules, which are produced from the breakdown of carbohydrates, fats, and proteins into carbon dioxide, water, and energy. Inhibitors of Krebs cycle are molecules that interfere with the normal functioning of the cycle. These inhibitors can be natural or synthetic compounds, and they can have a variety of effects on the cycle. Some of the important inhibitors of the Krebs cycle are citrate synthetase inhibitors. Citrate synthetase is an enzyme that catalyzes the first step of Krebs cycle. Inhibition of this enzyme can prevent the formation of citrate, which is an important intermediate in the cycle. Natural inhibitors of citrate synthetase include ATP and NADH, while synthetic inhibitors include compounds such as carboxymethyl 4-methyl 2-pentanone and benzyl succinic acid. Then aconitase inhibitors. Aconitase is an enzyme that catalyzes the conversion of citrate to isocitrate in the Krebs cycle. Inhibition of this enzyme can disrupt the normal flow of the cycle. Natural inhibitors of aconitase includes nitric oxide and reactive oxygen species ROS, while synthetic inhibitors include compounds such as fluoroacetate and arsenite. Isocitrate dehydrogenase inhibitors. Isocitrate dehydrogenase is an enzyme that catalyzes the conversion of isocitrate to alpha-ketoglutarate in the Krebs cycle. Inhibition of this enzyme can disrupt the normal flow of the cycle. Natural inhibitors of isocitrate dehydrogenase include ATP and NADH, while synthetic inhibitors include compounds such as 2-methyl citrate and oxalomalate. Then succinate dehydrogenase inhibitors. Succinate dehydrogenase is an enzyme that catalyzes the conversion of succinate to fumarate in the Krebs cycle. Inhibition of this enzyme can disrupt the normal flow of the cycle. Natural inhibitors of succinate dehydrogenase include ATP and malonate, while synthetic inhibitors include compounds such as carboxyl and thinolyl trifluoroacetone. The importance of these inhibitors lies in their potential to be used as therapeutic agents for various diseases. For example, inhibitors of the Krebs cycle have been studied as potential treatments for cancer, as cancer cells rely heavily on the cycle of energy production. In addition, some of these inhibitors have been used as pesticides and herbicides due to their ability to disrupt the normal metabolism of plants and insects. ETC inhibitor or uncoupler. The electron transport chain ETC is a critical metabolic pathway that takes place in the inner mitochondrial membrane of eukaryotic cells and in the plasma membrane of prokaryotic cells. It is responsible for generating the majority of ATP produced in aerobic respiration. Several inhibitors and uncoupling agents can modulate the ETC, and here are some of them explained, like rotenone. It is a natural inhibitor of complex 1 NADH dehydrogenase of the ETC. Retinone binds to complex 1, thereby blocking the electron transfer from NADH to ubiquinone, leading to a block in the electron transport chain and ultimately to a decrease in ATP production. Then antimycin A. It is a fungal antibiotic that inhibits the electron transfer from cytochrome B to cytochrome C1 in complex 3 of the ETC. Thus, it stops the electron transport chain, leading to decreased ATP production. Then cyanide. It is a potent inhibitor of cytochrome C oxidase complex 4 of the ETC. Cyanide binds to the heme group of cytochrome C oxidase, inhibiting the transfer of electrons from cytochrome C to oxygen, leading to a block in the electron transport chain and a decrease in ATP production. Then carbon monoxide. It is a toxic gas that binds to the heme group of cytochrome C oxidase complex 4 of the ETC. Carbon monoxide inhibits the transfer of electrons from cytochrome C to oxygen, leading to a block in the electron transport chain and ultimately to a decrease in ATP production. Then uncoupling agents. They are a group of compounds that can cause the uncoupling of electron transport and ATP synthesis. These agents create an alternative pathway for protons to re-enter the mitochondrial matrix, bypassing ATP synthase and dissipating the proton gradient. This results in the dissipation of proton motive force and a decrease in ATP synthesis. Examples of uncoupling agents include 2,4-dinitrophenol, carbonyl cyanide, M-chlorophenyl hydrazone, and permogenin. In conclusion, the ATC is a crucial metabolic pathway for generating ATP, and its modulation by inhibitors and uncoupling agents can have severe consequences on cellular energy production. Understanding the mechanism of action of these compounds can help in the development of new drugs for the treatment of several diseases. Next topic, lipid membrane fluidity. Lipids are a class of biomolecules that are characterized by their hydrophobic nature. They are one of the main components of biological membranes, which form the boundaries of cells and organelles. Membranes are composed of a phospholipid bilayer, which consists of two layers of phospholipids with their hydrophobic tails pointing inwards and their hydrophilic heads pointing outward. The fluidity of the membrane is an important property that determines its functionality. Here we will discuss the role of lipids in membrane fluidity. Role of lipids in membrane fluidity. The fluidity of the membrane is determined by the type of lipids that make up the membrane. There are three main types of lipids that contribute to membrane fluidity, like phospholipids. Phospholipids are the main building blocks of the membrane. They consist of hydrophilic head and a hydrophobic tail. The head is composed of a phosphate group and a glycerol molecule, while the tail is composed of two fatty acid chains. The length and saturation of the fatty acid chains determine the fluidity of the membrane. Longer and unsaturated fatty acid chains increase the fluidity of the membrane. Then cholesterol. Cholesterol is a lipid molecule that is present in animal cell membrane. It helps to regulate the fluidity of the membrane by filling in the gaps between phospholipid molecules. At high temperature, it reduces the fluidity of the membrane, while at low temperature, it increases the fluidity of the membrane. Then glycolipids. Glycolipids are lipids that have a carbohydrate group attached to them. They are found on the outer surface of the membrane and are involved in cell recognition. They also contribute to the fluidity of the membrane by interacting with each other molecule in the membrane. Now let's learn factors affecting membrane fluidity. Temperature. At high temperature, the membrane becomes more fluid due to the increased movement of phospholipids. At low temperatures, the membrane becomes more rigid due to the decreased movement of phospholipids. Then fatty acid chain length. Longer fatty acid chains increase the viscosity of the membrane, making it less fluid. Then fatty acid chain saturation. Saturated fatty acid chains pack tightly together, reducing the fluidity of the membrane. Then unsaturated fatty acid chains have kinks in their structure which prevent them from packing tightly together, increasing the fluidity of the membrane. Then importance of membrane fluidity. The fluidity of the membrane is an important property that determines its functionality. A membrane that is too rigid can limit the movement of molecules across the membrane, while a membrane that is too fluid can allow unwanted molecules to cross the membrane. The fluidity of the membrane is also important for the proper functioning of membrane-bound proteins, which require a certain degree of flexibility to function properly. So, in the human body, the fluidity of cell membranes is important for the functioning of ion channels, which allow ions to move in and out of cells. In bacteria, changes in membrane fluidity can affect the activity of enzymes involved in cell wall synthesis and other cellular processes. So, in conclusion, lipids play an important role in membrane fluidity, which is crucial for proper functioning of cells and organisms. Then WNT and Hedgehog signaling. WNT and Hedgehog signaling. WNT and Hedgehog signaling are two important signaling pathways involved in regulating various developmental processes and homeostasis in animals. WNT signaling pathway. The WNT signaling pathway is named after the wingless and INT1 proteins, which were originally discovered in fruit flies and mice, respectively. This pathway plays a critical role in the embryonic development, tissue regeneration, and maintenance of stem cells in adults. The pathway is initiated when WNT ligands bind to the frizzled receptor and co-receptor low density lipoprotein receptor related protein LRP on the cell surface. This leads to the activation of the misheveled ESH protein, which in turn activates catenin protein by preventing its degradation. The activated catenin translocates into the nucleus and activates the transcription of target genes such as cyclin D1, which promotes cell proliferation. Then hedgehog signaling pathway. The hedgehog signaling pathway is named after the hedgehog protein, which was originally discovered in fruit flies. This pathway plays a critical role in embryonic development, tissue regeneration, and maintenance of stem cells in adults. The pathway is initiated when the HH ligand binds to the patched receptor on the cell surface, leading to the release of the smoothened protein from inhibition by PTCH. The activated SMO protein initiates a signaling cascade that ultimately leads to the activation of the transcription factor GLI, which promotes the transcription of target genes such as cyclin D1, which promotes cell proliferation. Then importance of WNT and hedgehog signaling pathway. Developmental processes. The WNT and hedgehog signaling pathway are crucial for the proper development of various organs and tissues in animals, including the brain, limbs, and skin. Then homeostasis. This pathway also play an important role in maintaining homeostasis in adults by regulating cell proliferation, differentiation, and survival. Then disease. Dysregulation of these pathways has been linked to several diseases, including cancer, developmental disorders, and degenerative diseases. Then examples of WNT and hedgehog signaling pathways. Organs and tissues in animals, including the brain, limbs, and skin. Then homeostasis. These pathways also play an important role in maintaining homeostasis in adults by regulating cell proliferation, differentiation, and survival. Then diseases. Dysregulation of these pathways has been linked to several diseases, including cancer, developmental disorders, and degenerative diseases. Next, examples of WNT and hedgehog signaling pathways. The WNT signaling pathway is involved in the development of the anterior posterior axis in vertebrates and the regulation of hair follicle growth in mammals. The hedgehog signaling pathway is involved in the development of the limbs and the formation of the neural tube in vertebrates. Overall, the WNT and hedgehog signaling pathways are critical for the proper development and homeostasis of animals, and their dysregulation can lead to various diseases. Then signaling. Cell signaling is a process in which cells communicate with each other to control cellular activities. Signaling pathways involve a series of molecular events that start with the signaling molecule binding to a receptor on the cell surface and lead to a cascade of intracellular events that ultimately result in a cellular response. There are various types of cell signaling, including autocrine, paracrine, endocrine, and juxtacrine signaling. So, autocrine signaling occurs when a cell secretes a signaling molecule that binds to a receptor on its own surface, leading to changes within the cell. An example of autocrine signaling is seen in cancer cells that produce their own growth factors to stimulate their own growth. Then, paracrine signaling occurs when a signaling molecule is secreted by one cell and acts on nearby cells that have the appropriate receptor. A common example of paracrine signaling is seen in the immune system, where immune cells secrete cytokines to signal nearby cells to respond to an infection. Then endocrine signaling occurs when a signaling molecule is secreted by a cell and travels through the bloodstream to act on distant cells that have the appropriate receptor. Then hormones are an example of signaling molecules that act in endocrine signaling. Then juxtacrine signaling occurs when a signaling molecule is presented on the surface of one cell and interacts with the receptor on an adjacent cell. Juxtacrine signaling is important in developmental biology. So that's about endocrine signaling. Occurs when a signaling molecule is secreted by a cell and travels through the bloodstream to act on distant cells that have the appropriate receptor. Hormones are an example of signaling molecules that act in endocrine signaling. Then juxtacrine signaling occurs when a signaling molecule is presented on the surface of one cell and interacts with the receptor on an adjacent cell. Then juxtacrine signaling is important in developmental biology, such as during the formation of the nervous system. Cell signaling is essential for many biological processes, including development, growth, differentiation, and apoptosis. Defects in cell signaling pathways can lead to diseases such as cancer, neurodegenerative diseases, and metabolic disorders. Understanding cell signaling pathways and their dysregulation in diseases can help in the development of targeted therapies for the treatment of these diseases. For example, inhibitors of tyrosine kinase, which are important in many signaling pathways, have been developed as cancer therapeutics. Additionally, drugs that target the insulin signaling pathway are used in the treatment of diabetes. How can the insulin signaling pathway be targeted for the treatment of diabetes? Insulin signaling pathway can be targeted for the treatment of diabetes by using insulin analogs that can bind to insulin receptors and activate the downstream signaling pathway. Another approach is to inhibit the enzymes involved in the degradation of insulin, such as dipeptidyl peptidase 4 DPP4 inhibitors to increase insulin levels and promote glucose uptake. Additionally, targeting the downstream effectors of insulin signaling, such as glucose transporters and glycogen synthetase, can also be a potential strategy for treating diabetes. So, another approach is to inhibit the enzymes involved in the degradation of insulin, such as dipeptidyl peptidase 4 DPP4 inhibitors, to increase insulin levels and promote glucose uptake. Additionally, targeting the downstream effectors of insulin signaling, such as glucose transporters and glycogen synthetase, can also be a potential strategy for treating diabetes. Then, what is the role of WNT signaling pathway in cancer and how can it be targeted for cancer therapy? The WNT signaling pathway plays a crucial role in cancer by promoting cell proliferation, survival, and metastasis. Mutations in the genes encoding components of the WNT signaling pathways are frequently found in various cancers. Therefore, targeting this pathway has emerged as a promising strategy for cancer therapy. For example, monoclonal antibodies against WNT ligands such as vantictumab and ipafericet, vantictumab and ipafericet have been developed to block WNT signaling and inhibit cancer growth. Additionally, inhibitors of downstream effectors of WNT pathway, such as catenin and T cell factor lymphoid enhancer factor TCF or LEF, are being investigated as potential cancer therapies. How can the MAPK ERK signaling pathway are targeted for the treatment of cancer? The MAP kinase ERK signaling pathway plays a crucial role in cancer for promoting cell proliferation, survival, and metastasis. Therefore, targeting this pathway has emerged as a promising strategy for cancer therapy. Several small molecule inhibitors of MAPK ERK pathway components, such as MEK and ERK, have been developed and are being evaluated in clinical trials. Additionally, monoclonal antibodies against growth factor therapies receptors, such as epidermal growth factor receptor EGFR, can block the activation of MAPK ERK signaling and inhibit cancer growth. EGFRs. So, neurotransmitters are chemical messengers that are synthesized and released by neurons or nerve cells to communicate with other neurons, muscles, or glands. They play a crucial role in the functioning of the nerve system and are involved in a wide range of physiological processes such as movement, sensation, mood, and memory. There are several types of neurotransmitters, including acetylcholine. ACH is involved in muscle contraction, attention, learning, and memory. It is also involved in the autonomic nervous system, which controls involuntary functions such as heart rate and digestion. Then dopamine. Dopamine is involved in the reward and motivation systems of the brain. Then acetylcholine is involved in muscle contraction, attention, learning, and memory. It is also involved in the autonomic nervous system, which controls involuntary functions such as heart rate and digestion. Then dopamine is involved in the reward and motivation systems of the brain as well as movement and emotional response. Then serotonin. Serotonin is involved in mood regulation, sleep, and appetite. Then norepinephrine. Norepinephrine is involved in the flight or fight response, which prepares the body for stressful situations. It also plays a role in attention and mood. GABA. Gamma aminobutyric acid. GABA is an inhibitory neurotransmitter that helps to regulate brain activity and reduce anxiety. Then glutamate. Glutamate is an excitatory neurotransmitter that is involved in learning and memory. Then endorphins are involved in pain relief and feelings of pleasure and euphoria. Then neurotransmitters work by binding to specific receptors on the surface of target cells such as other neurons, muscle cells and gland cells. This binding triggers a series of biochemical events within the cell which ultimately leads to a response. So the balance of neurotransmitters in the brain is crucial for proper functioning. Imbalances in neurotransmitter levels have been implicated in a range of neurological and psychiatric disorders such as depression, anxiety, and Parkinson's disease. Drugs that target neurotransmitter systems such as antidepressants and antipsychotics are widely used in the treatment of these disorders. Next topic, cholera toxin or botulinum toxin. Cholera toxin and botulinum toxin are both examples of bacterial toxins that can cause severe illness in humans. First, we'll be learning about cholera toxin. Cholera toxin is produced by the bacterium Vibrio cholerae and is responsible for causing cholera, a severe and potentially deadly diarrheal disease. The toxin is composed of two subunits, the A subunit and the B subunit. The B subunit binds to specific receptors on the surface of intestinal cells, allowing the toxin to enter the cell. Once inside the cell, A subunit activates an enzyme called adenylate cyclase, which leads to an increase in the levels of cyclic AMP, CAMP in the cell. This increase in CAMP levels results in the secretion of large amounts of water and electrolytes into the intestinal lumen, leading to the characteristic watery diarrhea that is associated with cholera. So what's happening here? A subunit activates an enzyme called adenylate cyclase, which leads to an increase in the levels of cyclic AMP in the cell. This increase in cyclic AMP levels results in the secretion of large amount of water and electrolytes to intestinal lumen. Then botulinum toxin. Botulinum toxin is produced by the bacterium Clostridium botulinum and is responsible for causing botulism, a severe and potentially deadly parasitic illness. The toxin is composed of several subunits, each of which plays a role in its toxic effects. The toxin works by preventing the release of a neurotransmitter called acetylcholine, which is necessary for muscle contraction. Without acetylcholine, the affected muscles are unable to contract, leading to paralysis. Botulinum toxin is one of the most potent toxins known, and even small amounts of the toxin can cause severe illness. And both of these toxins are considered highly dangerous and require immediate medical attention if ingested or contracted. However, they also have important medical applications. For example, botulinum toxin is used medically in small doses to treat certain muscle and nerve disorders, as well as for cosmetic purposes such as reducing wrinkles. Cholera toxin has been studied for its potential as a vaccine adjuvant, which can enhance the immune response to vaccines. Then, hydropathy index. Hydropathy index is a numerical scale used to measure the relative hydrophobicity or hydrophilicity of amino acids, which can be used to predict the location of transmembrane domains in proteins. The index is based on the free energy change of transferring an amino acid residue from a hydrophobic solvent to water. The scale assigns a positive score to amino acids that are more hydrophobic, prefer to be in a non-polar environment, and a negative score to amino acids that are more hydrophilic, prefer to be in a polar environment. So what is hydropathy index? Hydropathy index is a numerical scale used to measure the relative hydrophobicity or hydrophilicity of amino acids, which can be used to predict the location of transmembrane domains in proteins. The index is based on the free energy change of transferring an amino acid residue from a hydrophobic solvent to water. The scale assigns a positive score to amino acids that are more hydrophobic, prefer to be in a non-polar environment, and a negative score to amino acids that are more hydrophilic, prefer to be in a polar environment. The index is typically represented as a graph with each amino acid plotted based on its hydropathy score. The most commonly used hydropathy index is the Kite-Doolittle index, which assigns a score to each amino acid based on the average free energy change of transferring the amino acid from a hydrophobic solvent to water over a range of different peptides and proteins. The hydropathy index is an important tool in protein structure prediction and can be used to predict the location of transmembrane domains in membrane proteins. Transmembrane domains are regions of proteins that span the cell membrane and are important for a variety of cellular processes such as signal transduction and ion transport. For example, if we consider the amino acid sequence of a protein and calculate the hydropathy index, we can predict which regions of the protein are more likely to be transmembrane domains. This information can be useful in understanding the function of the protein and designing experiments to study its role in cellular processes. Additionally, the hydropathy index can be used to compare the hydrophobicity of different amino acids and identify similarities and differences in protein structure among different organisms. Then aquaporin or GLUT types. Aquaporin and GLUT transporters are proteins involved in the transport of water and sugars respectively across cellular membranes. So aquaporins are a family of transmembrane channel proteins that facilitate the transport of water across cellular membranes. They play a crucial role in maintaining fluid homeostasis in various tissues and organs, including the kidney, lung, and brain. AQPs are found in all living organisms, including bacteria, plants, and animals. Some important points are there. Aquaporins are highly conserved and share a common structure with 6 transmembrane domains and 5 connecting loops. Aquaporins are selective for water molecules and their pores exclude ions and other small molecules. Aquaporins are regulated by various factors including phosphorylation, trafficking, and protein-protein interactions. There are 13 members of the AQP family in humans, each with a different tissue distribution and functional role. For example, AQP1 is found in erythrocytes, renal tubules, and endothelial cells, where it facilitates the rapid movement of water across the membrane. AQP2 is expressed in the collecting ducts of the kidney and plays a critical role in water reabsorption and urine concentration. AQP4 is abundant in astrocytes in the brain, where it helps to maintain the water balance and prevent cerebral edema. What is the importance of this? Aquaporins are essential for normal physiological functions in various organs and tissues. Aquaporins have been implicated in various pathophysiological conditions including edema, urinary tract disorders, and brain swelling. AQPs are potential targets for drug development, and AQP inhibitors and activators have been developed for the treatment of various diseases. Then GLUT transporters. GLUT transporters are a family of transmembrane proteins that facilitate the transport of glucose and other sugars across cellular membranes. They play a critical role in maintaining glucose homeostasis in various tissues, including the liver, muscle, and brain. Then some important points. There are 14 members of the GLUT family in humans, each with a different tissue distribution and functional role. GLUT transporters are facilitated diffusion carriers that transport glucose down its concentration gradient. The transport of glucose by GLUT transporters is regulated by various factors including insulin, AMPK, and protein-protein interactions. Examples: GLUT1 is expressed in various tissues including the brain, erythrocytes, and the blood-brain barrier. GLUT2 is expressed in the liver, pancreas, and small intestine and plays a critical role in glucose sensing. So GLUT1 is expressed in various tissues including the brain, erythrocytes, and the blood-brain barrier. Glucose sensing and regulation of insulin secretion. GLUT4 is expressed in adipose tissue and skeletal muscle and plays a critical role in insulin-mediated glucose uptake. What is importance? GLUT transporters are essential for normal physiological function in various organs and tissues. GLUT transporters have been implicated in various pathophysiological conditions including diabetes, obesity, and cancer. GLUT transporters are potential targets for drug development, and GLUT inhibitors and activators have been developed for the treatment of various diseases. Next topic we will be learning with the help of next video.